Hereby, I open this academic ceremony in which Chaitra Mandyam Kope Sarathy will defend the academic thesis, Genome-scale modeling of human adipocyte metabolism, strategies for multi-omics integration and visualization. May I invite you to present a summary of your study and the conclusions of your thesis?
Thank you, Prorector. I will now share my screen. Can you see my screen?
Yes, for me it works very well.
Good. Okay. Dear Prorector, dear committee members, dear friends, family, and audience. It's a pleasure to present to you in the next few minutes the research I've done towards my PhD thesis titled Genome-scale modeling of human adipocyte metabolism, strategies for multi-omics integration and visualization. A day in our life involves several activities that include both leisure as well as more intensive tasks such as exercising, working, and decision-making. All these activities operate on the currency of energy, and our bodies basically act like machines, such as cars, which convert petrol into energy that propels the wheels. Analogously, we convert the food we eat into energy for our daily activities. The process that drives our function is a complex interplay of chemical reactions, which is termed as metabolism. As you can see from the figure on the right, several chemical reactions occur within each cell of our body. You'll see figures like this in the rest of my presentation. The circles here represent molecules, and the arrows represent reactions. If you follow the arrows like so, you can trace the chemical paths of how input molecules are processed into output products. While the processes of metabolism are important for sustaining life, any dysfunction in these processes can affect the body adversely and cause disease. For example, imbalance in lipid metabolism can lead to increase in cholesterol. Excess accumulation of fat tissue leads to overweight and obesity, and impaired insulin release leads to diabetes mellitus. Many of these metabolic disorders are considered some of the most pressing concerns of the 21st century. Therefore, it is important to investigate metabolic imbalances to develop drug targets and treatment solutions. The overarching objective of my thesis, therefore, was to investigate molecular processes in obesity that occur in fat cells or adipocytes using mathematical modeling approaches. Mathematical models are used in biomedical research to simulate the physiological phenomena. They use differential equations to understand how the concentration of a molecule such as glucose changes with time. Here you see an example of 3 different molecules which behave very differently over time. Then the models can be tweaked to mimic the disease state and predict how the related chemical processes are affected. The biggest advantage of using models in biology is that they can be simulated on a computer. In most cases, these simulations require less time and much lower capital investment compared to running biological experiments in the lab. Modeling of biological systems began with building models for microorganisms such as E. coli. They're comparatively simpler systems to model, having about 3,000 reactions versus over 10,000 reactions in humans. However, due to the complicated nature of human metabolism, the methods used for modeling microorganisms are not directly translatable. Therefore, the currently available methods need to be adapted for studying human metabolic disorders, or new methods must be developed. In my PhD, we addressed some of the methodological challenges for human genome scale modeling. Specifically, we developed two methods to improve visualization and integration of experimental or omics data. And then we applied these methods in two studies to understand adipocyte metabolism. Now, I'll tell you about the first method that we developed, which is called Efmbis. So the set of metabolic reactions in the models can be seen as a map. Just like a city map on the left tells us how to get to one place to another, metabolic networks on the right can tell us where molecules go, the path that they take, and what they get converted into. Visualization of such routes can improve our understanding of where and how metabolic dysfunctions occur. But due to the complexity of human models, there was no straightforward way to visualize the pathways of interest. So we developed Efmbis to visualize the metabolic networks and map data. One of the benefits of Efmbis is that it needs minimal manual intervention. Here you see an example of a human pathway visualized using Efmbis. In this pathway, glucose, seen here, is being converted into lactate here. Circles represent molecules and small green squares are reactions. Genes associated with reactions can also be visualized, and they're shown by large squares like here. This is how the network looks like with and without mapping gene expression data. Looking at the color code for the genes, you can see that the genes in red over here are more active compared to the genes in blue over here. The biggest advantage of Efmbis workflow is that it is reusable and customizable. In addition, it can be used regardless of the size of your model. That is, Efmbis is applicable to both microbial and human models. To improve its accessibility to other researchers, the code for Efmbis has been integrated into Cobra Toolbox, which is a community standard software suite for the field of genome-scale modeling. Now, let me tell you about the first of our application studies where we applied Efmbis to investigate adipocyte metabolism. In our body, fat tissue is distributed across different organs. If we zoom in, we can see that the fat tissue is composed of fat cells or adipocytes, and there is blood supply to the adipocytes, which carry nutrients. When we zoom in further, we can see that as part of the metabolic processes, there is exchange of molecules between adipocytes and blood, which I'll refer to here as crosstalk. In this application study, we looked at a class of molecules called amino acids, which are increased in obesity. Our objective was to pinpoint how this increase could be caused by the imbalance in adipocyte metabolism. So to study this crosstalk, we mapped data from obese individuals generated in the Diogenes study onto an adipocyte model. Then we used Efmbis to visualize the dysfunctional pathways. In this figure, breakdown of isoleucine, an amino acid, has been visualized using Efmbis. This is a complicated figure, but I will walk you through it slowly. So analogous to the crosstalk that I described earlier, the whole process occurs in different parts, both inside and outside adipocytes. This is shown by the dotted lines here. The outside parts are blood vessel and the space between adipocyte and blood vessel. The inside parts are cytosol and mitochondria. I would like to draw your attention to the large squares throughout the pathway, which represent genes, like here. These squares contain bar plots, where the left bar indicates data from females and the right bar indicates the data from males. The color denotes either an increase or decrease of activity. If we look at the plots inside the mitochondrial genes, such as here and here, we can see that for most of the genes, there was low activity in females, as shown by the downward blue bars on the left side. Whereas the bars on the right side were either mostly flat and some are upward. This tells us that the pathway breaking down isoleucine, was different between males and females. Thus, by integrating data with model and visualizing them, we were able to derive novel insights into metabolic pathways. Next, we needed a method to compare two different conditions such as normal and disease. Therefore, we developed Comet, which is short for comparison of metabolic states. Comet can extract the pathways whose reaction rates function differently between the conditions. Now, let me tell you how Comet works. I mentioned earlier that the reactions in the models can be seen as a map or network. Mathematically, the rates of these reactions form a multidimensional space. Let's consider two states in blue and red. The data from these states is subjected to a series of transformation steps. The dimension is reduced and rotated. Then the reduced data are combined and signals are separated to extract common and unique aspects. Once these are mapped back to the network, we can clearly identify the pathways which are active or different in both the states. Comet can be applied in two ways. First, with simulated metabolic states to generate hypotheses that can be used to inform in planned laboratory experiments. Secondly, using the data that has already been acquired, as in the case of the Diogenes study. For our second application study, we used gene expression data from individuals with obesity that was acquired before and after weight loss. We then applied Comet to identify metabolic signatures and differences during weight loss. These are the reactions that Comet identified to be different, and they have been visualized here using Efmbis. So compared to the figures that I have shown you before, this network is different in the sense that the circles here actually represent entire reactions as opposed to just molecules. These reactions are connected if they share an input or output molecule. In this form of visualization, we can see how reactions interact across the network for the conditions being investigated. From the color code, we can see that the reactions in red, like here, were active before weight loss. Those in green, like here, were active after weight loss. Those in orange, like here, were modulated in both conditions. Thus, through this analysis, we were able to pinpoint changes in a subset of pathways in the large metabolic map. While I cannot go into details of each pathway in this presentation, we showed that Comet can be successfully applied to a real-world data set. In summary, the studies in my thesis represent methodological advances addressing different challenges for data integration with human genome scale models. Efmbis enabled visualization of metabolic networks and data mapping. Comet enabled data integration and comparing metabolic states. The application of these methods to the Diogenes dataset furthered our understanding of adipocyte metabolism in obesity. The methods demonstrated in my thesis can also be applied to other tissue models, such as liver and muscle, to study various other metabolic diseases. Hopefully, this can pave way for more biomedical application studies that will eventually improve patient outcomes. So with this, I thank you all for your attention, and I will now give the word back to the prorector.
Thank you very much for your presentation. Then we will slowly move back to the opposition. Yes. So the first opponent will be Professor Van Riel. He was a member of the assessment committee, and he also chair in biomedical systems biology at Eindhoven University of Technology in the Netherlands. Please, Professor Van Riel.
Thank you, Mr. Prorector. Dear candidate, it's a pleasure for me to open the opposition and to ask you the first question. And we have interacted for quite a while already, though not intensively. And I also want to congratulate you with your thesis, and I also want to include your 4 supervisors. So it's a big team of supervisors, so it should be good work. But let's see. Chapter 2, you discuss about elementary flux modes, EFMs. And that's where you also then build this visualization toolbox upon. However, later in your thesis, in particular in chapter 4, you say, well, EFMs are unsuitable for large genome-scale metabolic models. And also you say, well, it's I think that's you also state it's difficult to estimate the likelihood of observing an EFM in a given phenotype. So basically, there you discard, or you seem to discard EFMs. But so I'm happy to hear your opinion about how useful the concept of EFMs is in the context of genome-scale metabolic models.
Yeah, dear highly esteemed opponent, thank you for your kind words and questions. So yes, EFMs, I think they have found the most utility in the field of with microbial models to engineer microbes. So if one is interested to produce biofuels or certain therapeutic compounds, EFMs can be used to optimize the pathways that the microbes can take to produce in order to maximize the yield of the product. So in that sense, EFMs, I think, so far have found more applications with smaller models. But the problem with applying them on large models such as humans or human tissue models, it's computing the total number of EFMs would be computationally very intense. Also, the total number of EFMs that any such large model would have is also extremely large. So the studies that have tried to estimate EFMs from large models have done so by looking at certain subsets of the model or focusing on only a few subsystems, like in the case of cancer. So I think so they can be useful even with large models if one can, reduce the model to a certain subset of interest and then look at EFMs in the smaller subset.
Okay, thank you. My second question relates to your statement about objective function. Also in the chapter 4, in the same introduction, you also basically say, well, you say I prefer sampling-based methods because this does not require the specification of objective functions. So that seems to imply that you don't like objective functions. On the other hand, also in your presentation just now, you mentioned that one of the advantages of mathematical models, in fact, I think you called it the main advantage of models, that they can be simulated. And typically, in context of GEMS, simulation implies defining objective functions and solving FBA type of problems. So also here, what is would be your opinion about indeed the relevance of objective functions and FBA in the context of your work?
Yeah, so with objective functions and trying to find or pick one objective function for human tissue models, it's difficult because, say, for example, a stage is undergoing differentiation from a stem cell to a specific cell type. Then throughout the process of differentiation, the objective of the cell can be different. So it's very hard to pick an exact objective for models of such states. But in cases of microbial models or even cancer cells, the objective of maximizing growth has often been used. And in case of cancer, that would also work. But when we were looking at adipocyte, we were also, not sure whether maximizing, say, maximum fat storage would be ideal for, say, before weight loss or after weight loss. That is why we try to prefer methods which do not require specification of objective function. That way, the methods that we have developed can have more generalizable applications. A second thing that comes with these objective functions is also to do with the data on input uptake and secretion rates for metabolic models that is needed for FBA type of optimization. And this type of data is seldom available for human studies. So that way, having a non-objective function-based method, which require would be preferred for more widespread application.
Well, so apparently you interpret your statement about objective function that there is a single objective function for a certain cell type, cell-specific gem, and that this objective function should be a maximization of biomass formation. And I disagree in that sense, because I think, in particular for human tissues, you can actually specify many different objective functions. Basically, you already hint to that yourself. So there are examples of one of the earlier models developed, for example, for the liver, where actually I believe they tested about more than 400 different, let's say, metabolic tasks that they coded as objective functions. So, and I believe personally that running these simulations with objective functions representing different metabolic tasks is quite instrumental and actually essential to determine the validity of one's model. Because how can you otherwise know that your simulations, or at least your sampling-based solutions, have any physiological relevance? So could you comment on that? How did you check your physiological relevance of your let's say sampling-based solutions?
Yeah. So I agree that the advantage of the human models would be to pick a selection of metabolic tasks. But if the data to validate these assumptions, or the uptake and secretion rates are available, then after the simulation, we can go back and verify with experimental data. However, in our case, we did not have such information for the adipocyte model. So I agree that these need to be tested against experimental data. So it would be good to look for any publicly available data sets. But as far as my knowledge and literature search goes, I was not able to find such data to validate our simulations.
Yeah, I agree that if you would have, of course, the ultimate test would be matching it with experimental data, which is indeed difficult or almost impossible. I believe there is a midway out, so to say, where actually you can actually test the qualitative, let's say, capabilities of the model. I think that's also what was done for the liver, for the hepatocyte model. Can it do detoxification of alcohol? Can it do detoxification of ammonia? These type of tasks, and I think that is of course something what you could also investigate with an adipocyte model. I'm looking at the prorector. How are we doing in time?
Yeah, your time is up by now. So
okay. Then thank you very much, and I give the word back to the prorector.
Thank you, Professor Van Riel. And the opposition will now be continued by Professor Mariman. Professor Mariman was a member of the assessment committee, and he holds chair on functional genetics at Maastricht University. Please, Professor Mariman.
Yes, thank you, Mr. Prorector. Dear candidate, first of all, also my compliments for the contents of your thesis, and of course my compliments also to your promotion team. I think you did an impressive work with developing these methods and performing the applications. But still, I want to ask you a few questions, and that is about chapter 3. About the adipocyte model. And I want to go with you to figure 3.4. That's also the figure that you showed already in your introduction about the isoleucine catabolism. When I came to the end of the chapter, you mentioned that only the OR rule was used. And so I wonder if you would have used the AND rule also, or instead of OR, would that have made really significant differences for this particular metabolism, for instance?
Yeah. Dear highly esteemed opponent, thank you for your compliment and for your question. Yes, so the way to map data from the gene expression level onto reactions is in these models is through using gene to protein mapping information that is present in the models. For the when the adipocyte model, this adipocytes 1809 was developed, they have included only OR relations with for all the gene protein relationships. So what this would mean that they consider that the genes are either one of the genes is catalyzing that specific reaction, which of course I agree, which may not be the case always. So yeah, that could have a limitation on the analysis, but this is something that was out of the scope of our analysis. Our objective was to try to integrate data with the models and see if a pipeline like this might work. Of course, going forward, improving these GPR relationships with accurate and or relationships would be valuable to making meaningful interpretation.
So it would be nice to test that as well, I think.
Yes. Yes.
Let's look to the results in this figure, because apparently the mitochondrial metabolism genes are negatively associated with the BCAA levels in the plasma in women. But I see quite upregulation of the transporter genes. So then I start to wonder, does it mean that these amino acids are taken up by the adipocytes, but they are not processed? So where do they go? What do you think is happening here?
Yeah, so with the transport reactions from plasma all the way into cytosol, these are bidirectional transport. So these genes represent the activity around both uptake and secretion. So yes, the activity of these uptake and release enzymes are higher in females, showed positive association with plasma levels. But the mitochondrial genes had negative association. So when we look at the catabolic process itself, majority of the catabolism happens downstream of BCAT2, that is in the mitochondria. So BCAT2 and BK, I think DHB, those are the two critical enzymes for degradation. So what we think is happening here is that there is reduced breakdown of these metabolites in the adipocyte, and this incomplete breakdown is released back into the plasma. And this way, I mean, this is of course our speculation based on the analysis and results that we derived. But what we think could be happening is that because of a reduced breakdown, there is increased levels within the adipocytes, which is pushed out of the adipocytes and could be one of the factors for the increased levels.
So could these transporters be bidirectional? Could they export also or not?
Yeah. This is both export and import. Yeah.
Oh, all right. Then I can understand the situation here. You in that figure you mark these red reporter metabolites based on these gene data, and you say these are potential candidates for novel biomarkers. I want to ask, biomarkers for what, and what would you need to do to develop them into the clinical application then?
Yeah, so so these reporter metabolites show that there is significant transcriptional activity around them. And our idea behind identifying such reporters was to try to see how the information from the gene level is translated onto the metabolites. What I mean to say with the statement is that we can look at these specific reporter metabolites for further investigation. So the whether TCA, whether these metabolite levels, be it in this pathway or in the TCA cycle, whether they are linked to plasma levels, we do not know that. So these offer hypotheses to test in laboratory and actually study their relationship even further. And because this obesity has it's linked to insulin resistance downstream, these could also be candidates to maybe identify the onset of insulin resistance in adipose tissue, and this could be probably done by just metabolomics measurements from the tissue.
Yeah, that's okay. Yeah, you say they can be markers maybe for type 2 diabetes. Also in your introduction, you mentioned already the importance of the risk for developing insulin resistance in type 2 diabetes. However, when you selected your study cohort with people that have normal glucose levels, so would it not have been better to have a range of glucose levels from normal to increased to start your study?
Can you give a very brief answer, please?
Yeah. Okay. Yes, I think to specifically look at the relationship with insulin resistance, yeah, it would be good to have a cohort with stratify patients based on their glucose levels and look at differences in people with low glucose versus high glucose.
Okay, I thank you for your answers and give the word back to the product.
Thank you, Professor Mariman. The opposition will now be continued by Dr. Fleming. Dr. Fleming was a member of the assessment committee and is an associate professor at the National University of Ireland in Galway, and is also affiliated with Leiden University in the Netherlands. Please, Dr. Fleming.
Can you hear me?
Yes.
Congratulations, Ms. Sarathy. Congratulations on a fine thesis. I especially enjoyed reading the introduction chapter where you give a nice historical perspective on your work, and I commend you on looking back so far into the origins of the ideas that you ultimately are building upon. I have several questions, and they're connected together. So the main one I would have is thermodynamic feasibility and your simulations. The main questions are in that area. So you did check that your networks admitted a thermodynamically feasible flux. So that was good. But that doesn't exclude the existence of thermodynamically infeasible fluxes in the network, such as like cyclic cyclic fluxes that violate the second law of thermodynamics or energy conservation or both. So in your predictions, then your visualizations and your sampling, but in both cases there wasn't extra constraints applied or particular adjustments to the objective that might make sure that you avoid sampling or optimizing or finding an optimal solution that was thermodynamically infeasible. And I wonder, did this not worry you, or why you didn't consider this matter?
Yeah, dear esteemed opponent, thank you for your compliments and for your question. Yes, I think with the thermodynamic checks, I thank you for bringing it to our attention that using a different function might have been more useful to remove these cyclic roots. So how I see this might affect our networks is, removal of these cyclic reactions might reduce the space that we are processing with ICA and PCA. So that might, remove certain noise from the solution space itself, thereby improving the results that we would get. As far as the check with in the chapter 4 is concerned, I think that was a way. It was a proof of principle to test if pipeline like this can work on adipocyte data, and of course these were simulated metabolic states. So if there were cycles, and because they differ with say just 3 reactions, even if some infeasible cycles were present, it would be present in both of them. So it might just get cancelled out and as part of a common feature, if I can say so. So of course, removal of these cycles would improve our results and fine-tune the network modules to more biologically relevant one. I hope I have answered your question.
Yeah, so this is an example of a free lunch, it seems, in biological modeling, because indeed sampling both, even if they both had the same thermodynamically infeasible fluxes, when you take the difference, then they will disappear because they should be present equally in both. So that's the answer I was looking for. Then that brought to mind another question. So first you sampled, let's say, the control and the test model, and then you computed the principal components of both, and then you rotated both, and then you look for differences. But I wonder if you considered, instead of doing that, to take the 2 sample spaces, compute the difference, and then compute the principal components of the difference, and then rotate that. Would it give the same results, you think, or what would be the differences? Or maybe you did it already.
No, we did not test that, but we did consider doing the PCA or looking at just looking for differences in the sample space themselves, before doing the PCA and rotation. So what that would miss is that by doing PCA and rotation, we are picking out those reactions or reaction sets which have high contribution to determining the flux state of the model. So if we just look at the sample space, of course, we would find differences, but these may not be a strong contributor to the flux state, and their flux states, their changes in the reaction fluxes may not really or the results, I think, are fine-tuned because we are already looking for the key determinants and differences in them.
Okay. Switching gear now, perhaps more towards analytical chemistry, and in one of your chapter, I think it was chapter 5, chapter 3, you had selected 18 metabolites for targeted analysis. And I was curious, why those 18? What was the rationale for those 18? And what do you think about, the coverage of metabolism, just picking, let's say, 18 metabolites?
Yeah. So the measurements, these 18 metabolites were measured as part of the Diogenes study, and we were not involved in the decision about how or why these metabolites were chosen. So when I started the project, so my objective was to look for any relationship between these plasma metabolites and adipocyte metabolism. And we did realize early on that it's a very sparse data set, and maybe using FBA may not really work out. So we went about an elaborate route of just developing a workflow or a method that can work with just model simulation, and can integrate other omics data types. So to get back to the relationship, to studying the relationship, what we did was just to use the transcriptomics data set instead of the metabolites. And look for differences in before and after weight loss. And for looking at the relationship per se, we try to make a use of the network aspect of the models, and we try to identify the genes that are affected, metabolites inside the adipocytes that could be affected, and the pathways that could be affected.
Okay, thank you. Professor Peters, how are we doing on time?
Yeah, I think time is up for you.
Thank you.
Of the time left for the others. So thank you very much for your questions. Then the opposition will be continued by Dr. Van Grevenbroek, who is also a member of the assessment committee. And she's an associate professor at Maastricht University. So please, Dr. Grevenbroek.
Okay, thank you very much. Yeah, dear candidate, I also want to congratulate you with this very nice thesis. And of course, I want to also extend my congratulations to your promotion team. And I am not an expert in the field of modeling and systems biology, but I still enjoyed reading it. I learned some new things, and I think you've done a substantial amount of work. And as a first question, I first want to ask you about GEMs. And you indicate in your introduction that you that those GEMs are being constantly refined and updated. And I was just wondering, in the analysis as you were doing in your thesis, for instance, in chapter 3, how do you take into consideration genes that have currently unknown functions, because you have whole transcriptomics available?
Yeah, dear esteemed opponent, thank you for your compliments and for your question. Yes, the models are constantly being updated to accommodate for more gene functions that are discovered. But I think for the adipocyte model itself, as far as I know, this was the only well-curated model that is available. I know there is another one, but the model that I worked with had very good coverage of lipid-specific pathways, which is why we chose to use this model. But yes, I think this is a constant problem. I mean, it's not a problem, I would say. It's a feedback loop. The more we discover, the more we know about gene functions, we can always update the model. And the advantage of the methods, like the methods that I developed in my thesis, is that we can apply the same on the newer updated models and see how this would improve our results.
Yeah. Yeah, I see this. I think I see this a little bit, but I was also maybe a little bit concerned that somehow with these models, it seems that you sort of can get out what you put in. And that is also a little bit related to the question by Professor Fleming about the omics data that you used, and let's say the metabolomics. And you talked about, okay, 18 is not many, but actually from those 18, you chose to use only 3. So that sort of makes me also wonder, where does the term omics actually stop? Because if I measure 3 BCAAs just as single measurements in plasma, do I have an omics database?
Yeah. No, I completely agree that 3 would definitely not fall under the definition of omics. But I think that's why ideally it would have been good, I think, to have had metabolomics measurements from adipocyte to combine with the transcriptomics data set. But I would say that for the study in chapter 3, we only used the metabolite levels as a way to assess relationship between gene expression and the metabolite concentrations. Yeah. So yeah. Yeah, so I think in the omics in this then would be predominantly the transcriptomics data set.
Exactly, yeah, that's what I would think as well. And I was also wondering, those GEMs that you use, it seems to me that they are mainly focused on enzymatic reactions. Is that correct?
They're focused on the metabolic reactions. Yes.
Metabolic reactions.
Metabolic reactions.
Yes, but I was wondering, for instance, when there are processes such as, I don't know, receptor dimerization process, sorry, difficult word, but also, for instance, there is non-enzymatic glycation that can happen in cells, and that can also affect functions of, for instance, enzyme and hence metabolism. So how is that handled in these models?
So, so the coverage of reactions in these models is restricted to just the metabolic reactions. Of course, there could be, like you mentioned, influence on the enzyme level, whether there is an inhibition of an enzyme or activation of an enzyme after it's expressed. So, so I think there are efforts in the field now which are trying to incorporate such signaling information. But yeah, I think going forward, we should consider both metabolic and the signaling aspect to get a full picture of what's really happening within the cells.
Okay, thank you. And then I want to shortly go into your observation in chapter 3, in which you say you found sex-specific differences with respect to BCAA metabolism. And I really wondered exactly on which data did you base that conclusion?
So the sex-specific difference. So when we built this linear regression model to look for association between gene expression level and the plasma levels, so we found we did that to see, you know, in individuals with high plasma isoleucine, for example, are there certain set of genes which are also highly expressed or lowly expressed. So this linear regression model was the basis for determining whether sex is a significant factor. But
how did you determine whether it was a significant factor or not? That's what I was really curious about.
Yeah. So when we're building the model, we did look at different covariates like age, study center, BMI, fat percentage, and a couple of other things. So when we adjusted the model for sex, we saw a very significant reduction in the number of significant genes.
Yeah. But so usually, in general epidemiology, you would then call sex a confounder. And probably decide that you needed to adjust for it, rather than decide that there is sex differences in the actual relationship.
So, I mean, when we saw this significant reduction, we found that really interesting, and we decided to look at the male subset and the female subset separately.
Okay, I see. Yes, I guess my time is up.
I think so, yes.
And I give the word back to the prorector.
Okay, thank you very much, Dr. van Gevenbroek. And then we continue the opposition with Professor de Kock. He's also the secretary of this committee, and he also chair in population-based toxicogenomics. Please, Professor de Kock.
Thank you. So also my congratulations for this impressive amount of work with a high level of complexity on a challenging topic, which has also potential broad impact. So I will get back to this impact later on. But I think it's also written in very clear English, so my compliments on that. But still, it was not an easy read because of this complexity. And I didn't just read it, I really had to study it. And I think I spent more time than I do on average on the thesis. So that's a compliment also. So I have several questions, and I thought best to start at the beginning where the magic happens, which is at the methodology, which is chapter 2. So you describe a method to visualize elementary flux modes and the integration of different data types. And in figure 2.1, On page 30, you summarize this. And in panel A, you describe the inputs and you indicate gene statistics. Now, gene statistics can be pretty much anything. So my question is, do you mean specifically gene expression data, as you, for instance, use in case 2, or can it also be other types of gene statistics inputs or at different levels than individual genes, such as higher levels like pathways or processes?
Yeah, so dear highly esteemed opponent, thank you for your several compliments and for your questions. Yeah, so the workflow for Efmbis itself is quite flexible as to what data type can be mapped on the visualized network. So that is really dependent on the research question and the data that is available to the researchers. And in this workflow, I show relative fluxes and gene statistics as 2 examples of how data can be mapped on the networks. So with gene statistics, I did use different results from differential gene expression analysis for mapping on one of the networks. But if there is, say, gene correlation information like how we had in chapter 3, that can also be mapped onto the visualized network, and that is something we show in chapter 3 as well in one of the slides in my presentation. So I would like to say that because Cytoscape is the front end for the visualization and the network has metabolites, genes, and reactions, we can map omics data from one or each of these components onto a single network.
Yeah, so you could, for instance, also use epigenetic data like DNA methylation or microRNA data.
DNA methylation on the enzymes, you mean?
Yeah, if you would, yeah, you just said it works if you look at smaller subsets. So I could imagine that you have a research question that relates to the regulation of these gene expressions.
Yeah.
So for instance, if you would have DNA methylation information related to specific enzymes that you can map, it would also, yeah, I was wondering if that would be feasible.
I think in principle that should be possible, yes.
Yeah. So would it then also be feasible to sort of track down in your visualization at a later stage to see what effect in your outcome would be related to potentially even different types of data that you put in?
How do you mean to assess the impact of the DNA methylation? Or where?
Yeah, so for instance, so I was wondering if you have transcriptome data and regulatory data at the level of methylation, if you could visualize that separately, seeing where genes are upregulated and at the same time, so the expression, but at the same time are hyper or hypomethylated.
Yeah. I think this would require selection of, say, certain pathways where you expect to, after certain enrichment analysis, to pick out maybe one pathway or a few pathways of interest where you want to see if there is coordinated response between transcriptomics and epigenomics. So once maybe on top, from the top of my head, what I can say that if you would need to maybe pick or focus on some processes and visualize those processes as a pathway. And this is something we did show in chapter 3 too. After we did an enrichment, we picked out branching amino acid pathway and TCA cycle, but we have mapped data from the gene correlation and the reporter metabolites. But yeah, if there is the structure for such a pathway, we can map transcriptomics and epigenomics data.
Yeah, thank you. So then going to this use case number 2, which is actually on cancer patient data and for healthy and diseased tissue. I was wondering, because there's very little context on this data if you had any specific research hypothesis, or was it just to demonstrate that it works? And what was the selection criteria to use specifically this data set?
So I think so this was for the purpose of demonstration to show that visualization can be done from small microbial models and large human models. And I think the reason for specifically choosing this data set was also the compatibility between identifiers, the gene identifiers in the model and with the data set, where without any ID conversion, we had maximum coverage of the measured data.
Yeah, okay. Thank you. I want to quickly move on to your impact paragraph, which is something completely different, but because I see all sorts of possible applications for this, which we just discussed. But you're not very specific in what type of impacts you foresee. And in proposition number 9, you indicate that the methods you develop are instrumental in the development of personalized prevention. Now, how would you see that work in practice? What do you have in mind when you write this?
Yeah, so 2 things. I would say that the methods, both Efmbis and Comet, we have shown that it can be applied with a large clinical data set and to study a metabolic disorder like obesity. And to demonstrate that, we use the adipocyte model. I think if we had similar data sets, say from aging cohort in muscles, we could study that. And
I'm sorry to interrupt, but that's what you have written in your thesis, but that you can apply this. But how would that translate into a preventive strategy or a personalized preventive strategy?
I think
can you be very brief, please?
Yeah. So with the personalized medicine, I think we would maybe have try to have integration with machine learning approaches and more clinical parameters. So the more data that we have, then these machine learning algorithms could find patterns which are specific to individuals, and that way it can be taken further towards personalized medicine.
Okay, thank you. I give the word back to Professor Peters.
Thank you, Professor de Kock. Then we continue the opposition with the last opponent, that is Dr. Rieswijk, and she is a lecturer at Maastricht University. Please, Dr. Rieswijk.
Yes, thank you for asking the final question. I hope I still have enough time. So, dear candidates, congratulations, first of all, with writing this remarkable thesis. I found it really enjoyable to read, and yeah, I found it also very exciting to read that you focused a little bit on the FAIR principles, so making your data and your software more findable, accessible, interoperable, and reusable. And you say that you sort of face some difficulties also with relation to incorporating this. So maybe you could elaborate a little bit about what would be the ideal strategy for improving the fairness of your work.
Yeah, so dear esteemed opponent, thank you for your compliment and for your question. So the main challenge that I found with integrating in my work in general was finding the right data set to study a complicated condition like obesity or adipocyte function. So what I noticed is in most cases, the right type of data, like the metabolite uptake secretion, is not often available. And when in large studies such as Diogenes or other cohort studies, they might measure one omics data type, but it's not always available in the public domain. So I think the main challenge was in the findability of the data and the right type of data. So I would say that making it accessible or available upon request would be would improve would help us definitely, and that's where I see it going forward.
All right. Yeah, so you mentioned indeed that there's a difficulty in obtaining different data sets, and you used transcriptomics and metabolomics data, at least some limited metabolites. No, there's some question about whether it's called metabolomics or not. But do you think that's enough, or do you maybe need proteomics as well? I think Theo also mentioned a little bit about methylation data, microRNA data. Do you think that's where the future lies, or? And do you think it's reasonable to expect something like that?
I think, I mean, of course, large studies do have their own challenges with tracking a lot of patients over time. But, if the right amount of funding is available, I think it would be ideal to include at least metabolomics data set for gem-based analyses. But I would say that my comments about the data availability is from the perspective of, if I were to apply this method on any other public data set for a different research question, that would be the challenges. But I think, as far as more measurements is concerned, I think a little more depth of metabolite information would help.
All right. I think time is almost up, or I might still have 1 minute.
Or you can still ask a question as long as
okay. So so maybe really briefly, if you have time. So what do you think the impact of the role of the microbiome is in your work? I don't know if you can answer that quickly or not.
That's I think a broad question, but there are now models that have been developed for different microbiome.
Chaitra Mandyam Kope Sarathy, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. And I request that you and your company await the results of our deliberations and our return. So we will move from this stream and we will be back later. Chaitra Mandyam Kope Sarathy. The degree committee here present online has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Arts is authorized to confer upon you this academic distinction in accordance with Dutch university custom. And I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I do.
By the authority vested in us by law and in conformity with the decision of the committee here present online, I hereby confer upon you, Chaitra Mandyam Kope Sarathy, the degree of doctor, and grant you all rights attached by custom and law. As evidence of this, you will soon receive the degree certificate signed by the rector, the secretary and the supervisor, affixed with the official seal of the university, which is now shown by the beadle. Dear Dr. Mandyam Kope Sarathy, dear Chaitra, I have the honor to be the first to congratulate you with this great achievement. You did it.
Yes.
Congratulations also to your husband, Sriranga Kashyap, and other family and friends who are present all somewhere around the globe. It is a good habit to first thank the committee members that came from far. But in this case, traveling was of course not required. So also on behalf of my co-supervisors, Chris Evelo, Martina Sommer-Kutman, Marian Breuer, I nevertheless would like to very warmly and sincerely thank Dr. Ronan Fleming from the National University of Ireland in Galway, who is a true expert in the field, for critically reading the thesis and for being here today. I would also like to thank Professor Natal van Riel from Eindhoven University and our colleagues from Maastricht University, Professor Edwin Mariman, Dr. Marleen van Grevenbroek, Dr. Linda Rieswijk, and Professor Michel Dumontier, who was chair of the assessment committee but could not attend today. Chaitra, you are one of the original MaxBio PhD candidates. And you are in fact the last PhD candidate of SysTop to defend her thesis. That is not surprising because you actually started 1 year later than your colleagues. SysTop, which stands for Systems Biology Approach Towards Targeted Personalized Prevention of Chronic Diseases in the Obese System, was one of the funded original projects that kick-started MaxBio. And colleagues Chris Evelo, Thea de Kok, and Natal van Riel were involved from the very start of this project. When you applied for the position, you already lived in Maastricht because your husband had started as a PhD candidate at the Faculty of Psychology and Neuroscience. And we really felt lucky with such a perfect candidate. A background in biotechnology, so sufficient biological knowledge, lots of programming experience because you had worked in a large software company for a couple of years after completing your bachelor. And on top of that, you were already living in Maastricht. And we were not disappointed. During your PhD, you really developed complex methods to analyze omics data through data integration and visualization in the context of genome-scale metabolic models. And you also applied these methods to real-world data. For this, you used the Diogenes data set of the Maastricht Department of Human Biology. And Diogenes was generated more than 15 years ago, so hence the limited amount of metabolomics data that were available. But with your tools, you generated new biological insights from these data, which is really an excellent example of how reuse of existing data advances science. The two methods papers, where you present the tools that you called Efmbis and Comet, have been published in excellent journals. And both Efmbis and Comet are part of or are being considered for the COBRA toolbox. And as part of this toolbox, your methods will for sure be widely used and have large scientific and practical impact. On top of that, you designed really great logos for these tools, which can also be seen in your thesis and during the presentation today. And the biological interpretation of such complex biological data as you have worked with remains challenging, but your methods have definitely made it a little bit easier. During your PhD, you attended several conferences where you very successfully presented your work. You really enjoyed this, and you're also really good at it. You, in fact, won the Best Poster Award at ISMB 2020 in Canada, which was unfortunately an online event where you had to record a video to promote your poster. What you also really enjoyed was coffee at Banditos. And I hope you have found an equivalent of that in Toronto, where you are currently living. A PhD trajectory is never only easy. You had to deal with your initial daily supervisor, Michael Lenz, moving away. And this also led to slight readjustment of the topic of your thesis. Then you wanted to move to Canada because your husband had found a job there. As your supervisory team, we were okay with you finalizing your thesis from there, and then Corona hit. Countries were closed, visas were impossible to get. And luckily last summer, you were finally able to move. You rapidly finalized your thesis and found a great job, I believe. Chaitra, you've been a pleasure to work with over these past few years. You're a very nice person, you're precise, you're driven, but also modest. I really hope you have enjoyed this day so far and the discussions that were held. I hope you will have a great reminder of the day. You can be very proud of what you have achieved, and I wish you all the best in your future career. I give back the floor to the prorector now.
Thank you very much. Dear Dr. Sarathy, also on behalf of the Board of Deans and Maastricht University, I congratulate you with the honor you have acquired. And I would also like to thank all the members of the committee here present who have put effort into this defense and in reading the thesis. Thank you all very much. I hereby declare this ceremony to be ended.